Thank you very much. And thank you for the lovely introduction. So today, what I would like to start discussing with you is that we will base our discussions on some starting concepts, and that that a large proportion of mood disorders individuals, they are resultant of a deviant immune function caused by either an inborn or acquired monocyte and macrophage or T cell defect, which then will result in a malfunctioning or maldevelopment of the brain, an immune senescence, flares of chronic inflammation, and non-response to the current treatment. And what I would like to then show to you is how there is a role for the social determinants in leading to these maintenance, development and maintenance of the immune dysfunction that we see in these patients. So why study mental illnesses? I will concentrate most of my talk on talking about depression because most of my work has been done on this group of individuals. But from the literature and from some of my research, the disturbance of the immune system also applies to other severe mental illness such as schizophrenia and bipolar disorder. So major depressive disorder is a major global health issue. According to the World Health Organization, it affects around 300 million people, and it's one of the leading causes of disability worldwide. It's highly costly, both on an individual level and from the NHS, with antidepressants reported to be one of the most prescribed psychotropic drugs in England. Depression is very heterogeneous disease, which means that two individuals that may be diagnosed with depression, in addition to anhedonia, they may have some completely different symptoms. And interesting to me as a pharmacologist, and one of the reasons why I got involved in these, is that the current treatments that we have are only effective in about 30% of the individuals that take these drugs. So there's still a lot of room for us to improve the treatment that we deliver to our patients. So one thing that I would like us to think about in this talk is how the environmental stress, or the stress from the environment that we currently live in, they share some of the biological responses with the severe mental illness that we see. So stress and depression, they have both, they activate the sympathetic, the endocrine, and also the immune system. Inflammation may be related to depression on a two-way neuroimmune interaction. So glucocorticoid signaling via increased levels of cortisol inhibits the inflammation. But on the other hand, inflammation may impact brain function and brain functioning via changes on glucocorticoid signaling. And research over the last decade, and from my own group, shows how inflammation is associated with depressed patients and other mental illness patients, and may have a causal relationship with the etiology of depression. So what are the type of studies that link psychological stress with inflammation? So one of these studies that we conducted, so I conducted when I was an earlier postdoc, was what we call the Trier psychological stress test. And what we do here is in not necessarily patients, but just a normal healthy individuals, we ask them to our lab and we give them a short burst of stress. Sometimes it can be more of a cognitive stress, or sometimes it's just like a mathematics puzzle, or a psychosocial stress, asking them, for example, to speak in public. And during these stress, what we do is we collect different bloods on different time points. So we collect bloods before they start the stress test, in the middle, of this stress task and about 75 minutes after the stress has passed. And what is very clear is that in these individuals, there is an increase on these inflammatory response with markers similar that we see in individuals with severe mental illness, such as interleukin-6 and MCP1. So when we look at the blood of depressed patients, these markers are also there. So I did several different studies comparing the immune profile of individuals who are severely ill, such as treatment-resistant depressed patients, represented by the hash bars here, and compared them with healthy controls. And what you can see and appreciate is how inflammatory markers such as interleukin-6 and MCP1 is higher in individuals with treatment-resistant depression compared to healthy controls. And let's say the good cytokines that are responsible for the anti-inflammatory cytokines, such as interleukin-4, there was a trend to be decreased in these individuals. Not only cross-sectional studies show an association, but longitudinal studies also show that there is an association of inflammation with depression. So what we see here is that those individuals who present 2 occasions of high levels of C-reactive protein, which is an inflammatory marker, they have higher chances of developing depression 4 years later compared to those individuals who were not inflamed to start with, or even had just 1 occasion of inflammation. And when we do conduct Mendelian randomization studies, what we see is that these markers such as C-reactive protein and interleukin-6, they show the potential or a probable causal association with depression. So what my research after that points towards is that not only inflammation is associated with depression, but it points to a particular subtype of depression that it marks it as more severe. So inflammation was associated with a multi-system disruption, such as the disruption of the neuroendocrine. Inflammation was particularly associated with non-response to antidepressant treatment. Inflammation was associated with those who persisted years after years with the still increased depression, and in those who have depression comorbid with other physical illnesses such as coronary heart disease. Sorry, I think I've jumped 1 slide. There you go. So what we wanted to, although most studies concentrate on just looking at, a handful of inflammatory markers, we know that inflammation is a much bigger picture, and there's lots more of the systems involved. And therefore, what we wanted to do is we wanted to further investigate what is it really that the cell is doing, and what is happening in the cell when we say that there is an inflammation going on. So we did a high throughput technique that it's via Affymetrix profiling, and we looked at all 42,000 genes that were in which were differentially expressed between those with major depressive disorder compared to those with healthy controls. And what we did see that our sort of the key cytokines, such as interleukin-6 and TNF, they are here represented. Interleukin-1 is here represented. But in addition to these key cytokines that were validated, there is a large panorama of other markers that we see as disturbed compared to healthy controls. And what these shows is a wider picture of disturbance that it's not only on the immune system, but also on how the cells move and how the cells change their, the products within itself, and the way that they survive, and that the way that the cells communicate. So if you look at, of these cells, what they're doing, you can see that there is indeed a multi-system disruption. There's that is a much more complexity than just, one or two inflammatory markers. And when we look at what they are actually doing, is they are changing the way that cholesterol is able to support, how the energy system works in our cells. The inflammation has also been associated with how the cells are, if the cells are dying quicker, how these cells, they adhere and signal that the inflammation is occurring, and how it activates other cells to be on board. There's a problem with that as well. With how the cells, they just die quicker, they get disrupted, and so on and so forth. And by looking at how these disturbances, how do these associate with the clinical disruption that we see in our patients, we can see that that the cytokines have been associated with lots of the neurotransmitters that we know are associated with the symptoms that we see in our patients, such as interleukin-6, for example, has been associated with suicidal ideation, with aggression, with difficulty with fatigue and energy problems and motivation, sleep disturbances, and so on. So as I mentioned, what my research pointed is that inflammation seems to be being able to pick up those patients who currently do not respond to the most widely used monoaminergic drugs for depression. And therefore, the understanding of whether this is true, that maybe by measuring inflammation, can we actually pick up those who are unable to respond to treatment, will point to new directions. So what else can we then do? Which other anti-inflammatory drugs potentially can we then use to make our patients reduce their symptoms again? So looking at the treatment-resistant depression is important, because if you see the literature, you will see how debilitating these disorder is to patients. You will see that compared to those who do respond, the treatment-resistant depression, they have even higher frequencies of psychiatric and other comorbidities. They have even higher suicide attempts, even higher use of the primary care services, lower socioeconomic status, the higher BMI, and they tend to exercise less frequently, and they have a higher prevalence of psychosocial stress in their early life. So my interest in understanding inflammation was, well, is inflammation, can we subtype those who actually will predict the response, and is the inflammation a part of why the drugs do not respond to these patients? So the first question, is treatment-resistant depression associated with inflammation? Literature shows that indeed this is true. One of the largest meta-analysis, systematic review and meta-analysis, shows that there is an association of the pro-inflammatory cytokine interleukin-8 with response to antidepressants. And the other cytokine that has been consistently associated with treatment non-response is TNF levels. So responders, they showed significantly less levels of these particular cytokines than those who respond to antidepressant treatment. And this data was confirmed in other, in previous meta-analyses as well. So it's just been validated on 2 different meta-analyses. Longitudinal work conducted with the UK Biobank also shows an association of these the pro-inflammatory cytokine C-reactive protein with treatment-resistant depression. When looking at whether inflammation is also associated with other clinical predictors of treatment resistance, we can see that, yes, this is also the case. So clinical predictors of antidepressant non-response, such as obesity, such as early life stress, other medical illnesses, and some personality disorders or anxiety, have all been associated with inflammation in a smaller or wider degree. And we ourselves have looked at whether the different cellular profiles, so the number of cells, of the immune cells in depressed and who respond to treatment are different from those who do not respond to treatment. And we can see, and what we see is this pattern of immune senescence occurring in particular in those who do not respond to treatment, where they had lower levels of NK cells, so the natural killer cells, meaning that they will have a lower ability to combat any kind of infection, and they also had a higher levels of the cytotoxic T cells. So just showing that these immune defects, they also, they are also present in another T cell level. When investigating whether the treatment-resistant depression is associated with these with inflammation, we conducted a randomized control trial of venlafaxine versus imipramine. So here, and looked at our markers, so our inflammatory markers. So here, what I show you is this line represents healthy controls, and these are all individuals with depression. The first line represents people who were at baseline, so before getting any treatment, venlafaxine or imipramine. And the second part represents after 8 weeks of the antidepressant. So these are people who did not respond to antidepressants, either antidepressants, and these are those who did respond. So these are before the drug, so before antidepressants and after antidepressant. And what you can maybe appreciate is that even before antidepressants, there is already a difference between those who did not respond to treatment compared to those who did respond. And what you can see is that there's much more red, so much more inflammation going on in those who would not respond to treatment 8 weeks later. And after treatment, the individuals who did not respond, they continue inflamed. So you can see that the red relatively continues the same here, whereas in people who did respond to treatment, they did seem to decrease their inflammation, so they become a bit more green afterwards. And when conducting a Mendelian randomization studies to see whether these cytokines are indeed likely causal to the, to how the individuals respond to antidepressants or whether they achieve remission, again, we see a significant association with likely causal for both interleukin-6 and the interleukin-1 system, so the inflammasome system. So why could it be? What is it really happening that these cytokines are leading? Are the cytokines themselves leading to non-response? So the literature shows that these pro-inflammatory cytokines, they increase the expression of the monoamine reuptake inhibitors, and they decrease the monoamine synthesis. So there are several different ways by which these cytokines have shown to impact the way that these drugs work. One of which is by the production of the indolamine 2,3-D oxygenase. So if these cytokines, they increase these enzymes, what it means is that instead of tryptophan going towards the production of serotonin, and therefore being a beneficial for the neurotransmission in the brain, it goes to the kynurenine pathway and then to a more neurotoxic pathway, leading to the cells dying. There are different other ways that these cytokines have shown to impact and circumvent the mechanism of action of our conventional antidepressants. So, for example, cytokines have shown to increase this cofactor, decrease this cofactor, the BH4, which is an important cofactor on the production of serotonin and also L-DOPA, associated with the production of neurotransmitters. Cytokines, via increase in NF-kappa B and decrease in neurotrophic factors such as BDNF, they decrease how the neurogenesis, so how these cells, how our neuronal cells are able to make these synaptic connections and lead to higher cell death. And in addition, cytokines have also been shown to impact glutamatergic neurotransmission. So these cytokines, they decrease the glutamate reuptake and they will increase the glutamatergic release, and therefore, again, leading to excitotoxicity, so neuronal excitotoxicity. When we think about where this inflammation comes from, I think most of the work so far, most of the work so far points that it is, that there is a higher chance that inflammation may be associated with this stress, that the environmental stress that occurs in our daily life, and therefore there's a probability, there's a big probability that the social determinants of health impact the health outcomes, potentially via a change on these inflammatory markers. So what are the social determinants of health? Just so that we are all we know what we're talking about. So the social determinants of health, they are factors that influence our health equity in our daily living. They are aspects that will change, for example, income or social protection, unemployment or education, social discrimination, or even structural conflict or affordable to health services. And according to the WHO, the social determinants of health, they explain health outcomes on a much higher rate than genetics, for example. So social determinants, they explain up to 30 to 55% of health outcomes. And it's a part that it's very underlooked at the society and how we treat our patients so far. So there is still a lot that we can do in relation to this. And when thinking about inflammation, the social determinants are important because the social determinants have been associated with systemic inflammation over the life course. When thinking about where this systemic inflammation comes from, there are several different aspects of across the lifespan that lead to inflammatory changes, such as chronic infections, pollution, physical inactivity, whether a person is obese, whether there's a gut dysbiosis, a problem with the diet, chronic stress that I have already mentioned, sleep disturbances, or use of drugs. They all have been associated with this, with chronic inflammation that then has been associated as a trigger for different types of illness across the lifespan. So not only depression and other severe mental illnesses, but also cancer, cardiovascular disease, immunosenescence, neurodegenerative diseases, so on. In relation to the social determinants, most of the work has been conducted with childhood trauma. And what they have shown is that particularly individuals who have been through childhood trauma, they show this disturbance of the stress response that we have talked about, showing an activation of the hypothalamic pituitary adrenal axis with increased levels in cortisol and a change in the immune response. And the way that we currently understand is because our evolutionary speaking, our body needed ways to understand what's going on in the environment and protect ourselves from from physical injury or potential infection. So our body learned that as soon as there is an environmental stress going on, we should activate the inflammation so that our body would be ready to combat any bacteria or any other infection that could happen in case of wound, and increase the wound healing in case there was cellular disruption. And what we now see is that although these physical threat is not present any longer so often, let's say, any longer in our society, our body has a conserved transcriptional response. So our body still remains with this similar pattern. And nowadays, no matter whether the threat is a psychological threat, our body still responds on the same way, with the activation of the adrenaline and activation of cortisol, and leading to, and if it occurs long-term, leading to a disturbance of the immune system. So in response to trauma, with time, there is again a disturbance on these insufficient glucocorticoid signaling and insufficient gut dysbiosis, which then may contribute to the lifestyle, difficult lifestyle choices that some people have, so some behavioral mechanisms such as obesity, substance abuse, poor sleep, and other types of psychopathology. One way to understand this is a paper discussed by Bronfenbrenner, and which has been cited over 21,000 times. And what it does is that it puts our, the individual on a much broader system that I believe we need to think about. So when we think about everything that actually affects an individual, we need to think about the macro system that the person lives in. So, for example, what is the a predominant religion in the community, the amount of parks and green spaces and transport, the type of the ecosystem that would then involve the size or type of housing, or whether they live on a very crime bound neighborhood. The mesosystem would involve the amount of friendships or the extended family or relationships with the class teacher, and the microsystem, which then lead to the more family support, the nutrition, and all of these would then affect what is actually going on in our body. And we call these the bioexposome. So what we are exposed to it on a micro level or on a micro level, all will change how our genes are actually doing and which cells are expressed in our body. And what I would like you to think about is that the more social integration one may feel, the more intimate relationships and the higher levels of cultural resources and friendship, this will then decrease how the threat and give us a higher sense of security. And in these cases, then the individual has a lower stress response. And if these things are not in place on our, on our exposome, so what we are exposed overall, what happens is that there is this feeling of threat that, that things are not okay. And therefore, our body then consciously or unconsciously will generate cortisol that will then, with time, might impact the immune cells and impact also our microbiome that may then lead to changes on our brain function. So the social environment has a big role on how our, health and behavior, on our health and behavior. So the social environment, because of its changes and because of the way that we are exposed, by the physical or the built environment, the chemicals, the physical, chemicals and microbes that we are exposed on our environment, they may change the way that our system responds. And similarly, the social environment, by giving us a sense of threat or a sense of security, may induce the brain to produce these hormones that will then cause our cells to receive the signal transduction and lead to the production of proteins that change our health and behavior. And nowadays, this system is called the social signal transduction. So where what it discusses is how the adverse environment, by the social determinants that we were talking about, such as the instability or the feelings of loss and trauma, they are able to change how our brain works by this perception of threat or safety and security that would then lead to an increase in the neuroendocrine signaling, such as cortisol or adrenaline, that would then impact our cell transduction with these more molecular transcriptional factors that eventually would lead our genes to express a certain molecules, which we now call the conservative transcription response to adversity. And this has been associated with health. And this is not something that it's stable. So this is something that there is actually, there's a change. So it is possible that with time, if someone feels a bit more safe and secure, then this system is not as much activated, and therefore there is, less triggers to the system, and less chances of impacting their mental illness. However, if anything changes in our social environment, that suddenly we now perceive our social environment as a more threatening experience, we can, the body would then change the way that these genes are expressed, and therefore could lead to different outcomes. But interestingly, there is a role for social support that I would like to discuss here. So at the same time that loneliness has been associated with a lower response to vaccine, if you improve a marital harmony, or if one improves the social network or the social support system, you can change how your immune system responds. And this may be associated with a better outcome for our patients. We ourselves are conducting a randomized control trial of physical exercise in patients with depression to further investigate the link with the environment, the social support, with inflammation. And in this case, what we would, the hypothesis is that by lowering inflammation, exercise improves cognitive function and motivation, and this would then lead to decrease in symptoms of depression. So our hypothesis is that in particular with those who have immune defects, exercise would be particularly beneficial because they would reduce inflammation, correct these immune disturbances, normalize the cortisol axis, normalize the immune system, and help individuals recover from their depression. And with that, I would like to thank you all people in my lab and my current collaborators, and any questions will be welcome. Thank you.